Greetings, ladies and gentlemen. Professor Recore here, and today we're going to do a question and answer session on histology and cell biology, particularly regarding the human digestive system. So let's get started. In this first question, we're looking at a histology tissue sample that was taken from a man who recently died of cancer, and we're asked to identify what part of the GI tract that tissue sample was taken from. In order to do that, we're going to identify what's there and what's not there in the image. So let's start first with what's there. The first big thing that we see in this image is the presence of multiple villi, villus as singular, and that right there represents one villus. Inside this villus, we have the presence of goblet cells, which secrete mucus. There's one right there, one right there. And then thirdly, we have the presence of a structure here in the middle that we're going to get into in the very next question, and I don't want to give it away just yet. Lastly, our epithelium type is simple columnar. So that's what's there. So what can we rule out just by knowing what's there? Well, the fact that we have villi and microvilli present allows us to rule out both the appendix as well as the large intestine as potential answers. The fact that we've got a simple columnar epithelium and not a stratified squamous epithelium allows us to rule out the possibility that the esophagus is the location that the tissue was taken from. Finally, the fact that we don't see any gastric pits present, as well as the fact that there's abundant villi and microvilli, allows us to rule out that the stomach was the source of the tissue. This allows us to narrow all the way down to our answer, which is the small intestine. In fact, the jejunum of the small intestine specifically. This next question is a follow-on from the previous question. And it says, examination of the lamina propria of the organ identified in question 3, which we can see below, reveals which of the following key histologic features of the GI tract. What I want to focus your attention in on is this area right here in the middle. So we know we're in the small intestine, and I told you that we were in the jejunum. And there are tubes of lymph in the small intestine that are responsible for absorbing lipids. And what are these tubes of lymph called? Lacteals. On this next question, we're asked to take a look at the histology in front of us and to determine from which portion of the GI tract was this slide taken. Right away, you see the presence of multiple gastric pits, which lets us know absolutely that we're looking inside the stomach. Here we have another question asking us to identify the location of the GI tract based on the histology. In the image we see below, one thing jumps out at us right away, and that is that there is a marked epithelial change that occurs over on the right-hand side of the image. And what we see on the right-hand portion is that we start off with stratified squamous epithelium, and as we move to the left, the epithelium changes to simple columnar. This is because the stratified squamous epithelium represents the most distal portions of the esophagus as it transitions into the cardiac portion of the stomach, and the epithelium becomes simple columnar. To confirm that we're in the stomach, we see the presence of gastric pits here on the left-hand side. That confirms for us that this histology represents our gastroesophageal junction. While this stratified squamous to simple columnar epithelial change is characteristic of the gastroesophageal junction, it's not unique in the GI tract. There is one other location that has a similar change, and that's at the recto-anal junction. At the recto-anal junction, however, going from proximal to distal, our epithelium changes from simple columnar to stratified squamous. And as we get distally in the anal canal, the stratified squamous epithelium becomes keratinized. But some of the things stand out about that allow us to differentiate it from the gastroesophageal junction. First of all, the simple columnar epithelium of the rectum doesn't have gastric pits. Moreover, at that junction, at the recto-anal junction, there are scattered lymphatic nodules and there are intestinal glands from the large intestine in the rectum at the portion where there's simple columnar epithelium. I just wanted to make sure I mentioned that one other instance in which there is a simple columnar to stratified squamous or vice versa change. However, there are noted differences, as I just mentioned. In this question, we're being told that we're looking at tissue taken from the large intestine, and we're being asked to identify the layer that's indicated by the double arrow. Well, right away, we can go ahead and say that we're looking at the muscularis externa layer. What's unique about the muscularis externa in the large intestine is that it's composed of two layers. The inner layer, which is the circular layer, and the outer layer, which is the longitudinal layer. The names circular and longitudinal are a reference to how the muscle fibers are arranged in each of those layers. The outer longitudinal layer in the colon or the large intestine is actually condensed into these three equally spaced bands that are referred to as the taenia coli. Our last question has us looking at a segment of the GI tract of a recently deceased woman, and we're asked to identify the portion of the GI tract that we see based on the histology. Jumping out at us right away are these large aggregates of nodular lymphatic tissue that we see right in the middle. These are known as Peyer's patches. Peyer's patches are particularly important for immune surveillance in the small intestine, and indeed the pale staining regions of these aggregates are the sites of B cell proliferation and activation. Peyer's patches are characteristic of that final portion of the small intestine that's known as the ileum. So that wraps up the histology and cell biology question and answer session regarding the human digestive system by Professor Recore. I thank you for joining me, and please leave a comment and ask any questions that you might have, and I'll see you next time.